Hi guys, this is Sayed and my group partner is Fatma. We are senior in Westfield State University and today we are conducted our research in ABA's response to drought in a species of plant called Arabidopsis. ABA is abscisic acid and basically that related with their response to stomatal guard cells regulation. Here we show the ABA mechanism. This slide we can see that it shows the ABA mechanism. During the drought, ABA signaling increases, which will activate the receptors on cells, forming the complexes. These will interact with proteins called PP2Cs, the relief effect of which is the downstream kinase. This will control the negative sign into the cells. We use the Here we use our research question, which genes affect the ABA signaling pathway and how do their effect play a role in Arabidopsis thaliana's response to an abiotic stress like drought. So our hypothesis is that we are expecting to see a direct correlation between a drought and ABA levels in Arabidopsis. That is, a high level of ABA will be present as a drought response, and that the genes correlated with ABA signaling pathway will be upregulated to account for this stress. So here I'm gonna explain you mechanism. So what we did, first we use reference genomes uh, to get SSR number from the papers. And second thing we did, after getting the SR number, we uploading this into the Cybers. This is a program and did run RMTA analysis. After that, we did R analysis was performed by using RStudio DSEC2 and obtained volcano curve. 9 genes were analyzed by using the TR data. From here, Fatma will gonna explain you about it. Thank you.
Hello, this is Fatima, and I'm here going to be sharing the results. Now, we included a volcano plot here as a visual of how many genes are being deregulated, upregulated, and genes that are not affected at all. We also included an index table of the 9 genes that we focused on. Most importantly here is the fact that those genes are being expressed in the same area or the same location, which suggests that the coping mechanism of this abiotic stress is happening at a very early stage, keeping in mind where they're being expressed in the roots, the seeds. Those are first responders of drought. Now, to the discussion, we see that there's an upregulation of PP2C. Now, this upregulation that's found in the differential gene expression data, it suggests that it's being abundant and interacts with the ABA complexes in the ABA signaling pathway. This increase in the PP2Cs actually causes a deregulation of a gene called OST1, and the inactivation of this gene causes an activation of SLAC1, which causes stomatal closure. Similar to SLAC1, we see SNRT2 found in high dispersal rate and upregulated. Another gene is SIR34, highly abundant and interacts with the protein very similar to PP2C, inducing the biosynthesis of abscisic acid piles. 6 and 4 showed deregulation. There are negative regulators at the ABA signaling pathway, so it makes sense that they're being deregulated. Arab1 is upregulated. It's a positive regulator of the ABA signaling pathway. Again, it makes sense that it's being upregulated. We compared our data to the reference paper that we used, and we saw that there's a lot of similarities between our data and their results, according to what we wrote over here. The most important part is this top one. WRKey54 is a kind of a complex that's very similar to Arab1, but it's the opposite, which means that because WRKey 5470 is a negative regulator, Arab1 is a positive regulator. It's a transcription factor that's a positive regulator, like I mentioned before. Now, all of these 9 genes that I discussed are further supported by a reference or research done by Yesneri and their colleagues in 2005. This is the results. Thank you.